Hi, everyone. Thank you for joining our webinar today, titled Solving Nanotechnology Application Challenges with the Versatile and Scalable Microfluidizer Technology. With me is Dr. Yang Su, the Manager of New Technology and Applications at Microfluidics International Corporation. Dr. Su holds a PhD degree in Chemical and Environmental Engineering from the University of Toledo. His current focus is to deliver the desired outcome for each customer's unique application using Microfluidics' cutting-edge microfluidizer technology. So my name is Julie Chen. I am your facilitator. We will have a Q&A session at the end. So without further ado, Young, I think I can go ahead and pass the ball to you. Feel free to share your screen.
Yeah, thanks for the introduction, Julie. And hi, everyone. My name is Yang Su. Thanks again for joining us today. As Julie has mentioned about the title of my presentation, I'm glad to have this opportunity to introduce you our microfluidizer technology and demonstrate how it can be applied in a variety of nanotechnology applications. Here's the agenda of my today's presentation. I'd like to start with a brief introduction about Microfluidics, which is my company, and then introduce you the microfluidizer technology. I'll briefly discuss some of the considerations and challenges that are commonly encountered during the manufacturing process for the nanotechnology applications, and explain some of the benefits of uniform processing before switch back to Julie for a quick overview or introduction for particle characterization technologies. Then I'll continue walk you through with several examples of demonstrating the technology in different applications across different industrial sectors, including biopharma, cosmetic, high-performance energy storage, as well as nutraceutical. I will say here that the microfluidizer technology is a proven and well-known technology for especially biopharmaceutical industries. I actually have covered a lot of that during my previous webinar with HORIBA, and also we have published a lot of good data and case studies. So I won't spend too much time on this topic today. If you're interested, feel free to read more, reach out to us. We'll try to focus a bit more on the other areas today. Before I end my presentation, I will quickly show you the diverse product portfolio we have at Microfluidics before we enter the Q&A session. So who is Microfluidics? Microfluidics as a company was founded 40 years ago, and we're specialized in producing high shear fluid processors using our proprietary interaction chamber technology. Interaction chamber technology is also our core technology. We're located in Massachusetts, just outside of Boston, but we have a global presence. We have sold our processors to many customers all over the world. About a little more than 10 years ago, Microfluidics was acquired by our current parent company, IDEX Corporation, and we joined IDEX material processing platform, or MPT platform, with a few other sister companies. Some of them you may have heard of, including Fitzpatrick, Metcon, Quadro Engineering, and Steridose. Microfluidics, as I mentioned, we specialize in producing the microfluidizer processor, which are widely used in both research and development, all the way to commercial manufacturing processes. And our technology can be used for a variety of industries, as I mentioned, specifically pharmaceutical and biotechnology industries, where we help producing pharmaceutical ingredients, vaccines, and our technology is also used in other industries such as chemical industries. We can be used for processing inkjet inks, coatings, and as well as for nutraceutical and cosmetic industries. We have a lot of experience working with our customers on the both application side and the machine designs. With our expertise, we ensure our customers have the right tool and the right support to help them to achieve their specific processing goals. Here's just a quick snapshot for some of the applications that works very well with our technology, including several of the lipid-based systems, I would say nanoemulsions, lipid nanoparticles, liposomes, and also polymer nanoparticles, or reducing polysaccharide molecular weight. The technology also works very well with dispersions which contains the solid, including cell disruptions and various deagglomeration and particle size reduction applications. Here's a quick example to show you that we have a complete product line from small lab scale to pilot and all the way to production scale. And with our excellent scalability, all of the system here, regardless of the scale, use the same fixed geometry interaction chamber design, which I will explain in a minute. So this is a quick schematic to show you the principle of the technology. Essentially, a product with large particles enters the system via the inlet. It can be an inlet reservoir, and the product will be pressurized by a constant pressure intensifier pump to pressures up to 30,000 psi, which is our maximum processing pressure. And there's such high pressure, the materials will be pushed through the fixed geometry interaction chamber. The interaction chamber has the microchannels in it, where the product will experience consistent and uniform high shear processing to achieve the desired process outcome. After processing or exiting the interaction chamber, the product temperature can be efficiently controlled via a temperature controlling device. It can be a cooling coil on a small scale or a heat exchanger on a larger scale if needed. We have two different interaction chamber designs, as shown on the graph is to the right. The top one showed the Y-type chamber. So in the case of the Y-type chamber, the product actually will be split into two streams inside the chamber, and each will pass through a microchannel. So the product will be subjected to high shear processing. And once it exits from the channels, it will form a confined impingement where intensive micromixing will occur. This in particular helps reduce the particle size for the liquid-liquid dispersion system, including nanoemulsions, liposomes, lipid nanoparticles, and polymer nanoparticles, and some other similar systems. The graph shown down below here is the Z-type chamber. As you can see, the Z chamber only has one microchannel, and the product will impact on the solid surface before exiting the chamber. The design of the Z-type chamber is more ideal for processing samples contain solid, including cell disruptions and particle size reductions and deagglomeration applications. So the advantage of our system or our technology paired with the fixed geometry interaction chamber is we can truly deliver a constant processing to achieve uniform processing results, as compared to some conventional technologies such as a valve-based homogenizer. In the case of a valve-based homogenizer, usually a homogenizer valve is used. The process pressure is typically controlled by adjusting the gap between the valve and the valve seat, and the valve is held by a spring. So the difference between the system is, in the case of Microfluidizer technology, we utilize a constant pressure intensifier pump combined with the interaction chamber. There's no moving parts. That's we can achieve a constant pressure profile during processing, as you can see from here. As a green curve present the actual process pressure from one of our system. When the intensifier advances to push the to pressurize the material, you can see the pressure ramp ups very quickly, and we'll be able to maintain that process pressure during the majority of that cycle. So the pressure drops down to 0 is when the intensifier pump retracts and get ready for the next push. And during this period of time, nothing gets processed. So even though the pressure is 0, it won't affect the processing results. The majority of the material will be processed at a constant pressure. If you use a valve-based homogenizer, the system is designed with a constant volume principle. So the pump will continue to pump the same volume regardless of what happens at the valve end. So what happens over there is the continual pushing of the pump will cause tiny vibration. So this is exaggerated here in this animation. But that tiny vibration can actually introduce a huge variation in the process pressure. You can see, go back to this plot here, you can see from the red curve, that is the pressure from a valve-based homogenizer. You can see the pressure can vary or change as much as 50% of the set pressure. If in this case 30,000 psi is the desired pressure, the pressure actually varies, dropped all the way down to 15,000 psi, which is half of the set pressure, and the material will actually experience or be processed at all of the pressures between different pressures between 30 and 15,000 psi. And if you calculate or convert the pressure to a theoretical shear rate profile, you can see we are able to process materials at a constant shear. And similar to the pressure profile, the homogenizer will process the sample at various different shear rates. So that indicate the process is not uniform. So first, the efficiency will be not as good because you're not processing majority of the product at the target shear rate level, and also it's not uniform process. Your results will be a more heterogeneous or with a wider distribution after processing. The second advantage is for the microfluidizer technology is its scalability. So we guarantee our scale up results. When we scale up, we don't follow the similar principle when the homogenizer scales up. In the homogenizer's case, you have to use a bigger valve design, so the geometry is bigger. The gap between the valve and the valve seat are bigger now to allow higher throughput of the product, and that obviously changes all the processing characteristics you have developed from a smaller scale, because now everything is different. The geometries are different as well. So in our case, we fixed the dimensions of the microchannel. Instead of making a larger channel, we actually use a parallel array of identical channels. So by that way, as long as the other process parameters are kept the same, for example, the pressure, every single channel will achieve identical shear rate and processing of the material. That's how we ensure that the scale-up results will match the lab scale results without any issues. You don't have to, once you optimize your process, you can directly scale up to the larger scale system. Now, just a quick slide to discuss a little bit about some of the considerations during manufacturing processes. I would say when you process material for nanotechnology applications, obviously the first goal is to achieve target properties, including different physical chemical properties. There are many properties depending on the application and the formulation and the materials being processed. But I would say in many, if not all of the cases, in many cases, particle size and distribution is probably one of the most important properties. Obviously, there are other important properties. I think I remember from the pool question, people are mostly interested in particle size measurement as well. So obviously, you want your processing method or choice of the equipment can not only deliver that target particle size and distribution, but achieve that very efficiently. Then you would consider the choice for processing equipment is it reliable. It should deliver repeatable results with batch-to-batch consistency, and it should be also somewhat future-proof if you need to scale up from your lab scale to production scale. The technology, as I mentioned, should offer a seamless scalability to a larger scale manufacturing. And certain processes, especially I would say for a lot of biopharma industry applications, the manufacturing process also sometimes can add additional benefit to allow easier downstream processes. One good example here is the sterile filtration process. In many pharmaceutical products for parenteral delivery or injectable drugs, the product needs to be sterilized before it can be administered. So for sterilization process, a very common and economical way is to use terminal filter sterilization. So a sterile grade filter is used to pass through the product and capture the bacteria. So to do that, the filters are made with membranes, have an average pore size of about 0.22 microns or 220 nanometers. So you can imagine the majority of your particle population should be less than this size to ensure a good, smooth, and efficient filtration process. Once you hit that particle size, get a good filtration process, which means you're also going to save a lot on the cost, not only by reducing the loss of the material, because any larger particles will be captured, and also less filter surface area or filter material will be required. So that's a win-win if you can achieve that good particle size and with tight distribution. Last but not least, some of the processes or the processing equipment should allow the process to meet regulatory requirements. For example, pharmaceutical processes needs to meet the GMP requirement, and I know there's certain other requirements for food industry and some other industries. So that's another consideration there. Now, if you can achieve uniform processing, you can imagine there are many advantages or benefits you can achieve from there. I listed a few here. By no means it's a complete list, but obviously you can see if you can achieve a uniform particle size, it will help increase product stability. And often that stability will lead to longer shelf life for some of the products. And for other certain products, it's important to achieve desired or even improve the appearance and feel of the products. Not to mention, if the process is efficient, it's going to save a lot on the manufacturing side. So it helps not just saving energy, but overall, I think it's going to help save the cost for manufacturing the product. And with that, I'd like to switch back to Julie for a moment and have her talk about particle characterizations.
All right, awesome. So really what comes hand in hand with microfluidizing technology is the need to have some sort of method in place that will help you understand, will let you monitor, how uniform or how small the particles have been processed. This is the prerequisite to achieve the desired result. And so, for example, looking at this chocolate tart right here, the size and size distribution of the chocolate particles will govern the texture or the mouthfeel for the consumer. So here at HORIBA, we've done a lot of studies with various different vendors that we know anything above 30 micron will be perceived as gritty and low grade. And so when you sell your chocolate, you then won't be able to charge a premium. So that kind of impacts your profit as well. And anything below 30 micron is kind of perceived as gritty, greasy. So not smaller is not necessarily better. It is very important to process this chocolate and know exactly when to stop using a particle characterization technique. And likewise, if we were to shift towards more of a life science space, like Young had mentioned, lipid nanoparticles, liposome, or vaccine manufacturing, the size and concentration of your viral particles will directly influence the bioavailability, or AKA efficacy. So for instance, your COVID vaccine, what is the load in a shot as it enters into our body, or after it's being reconstituted and placed by the window, on the table, does it remain stable and for how long? So these are all information that we can get just by looking at the particles. And that same goes with the coffee flavor, flavorant, or battery, or stability of your emulsions, and various different techniques. So that's what we do best here at HORIBA. We offer a suite of different technologies to help you understand your particles. So there's the laser diffraction, there's the dynamic light scattering, there is the nanoparticle tracking analysis, the traditional imaging analysis that you may be more familiar with, such as microscopy, sedimentation. Today, I think this case studies that you will see as I pass the ball back to Young are done using the LA960 laser diffraction particle size analyzer, which is the first instrument that you see. Laser diffraction is a very high throughput technique that kind of gives you a measurement result well within the 60 seconds. The LA960 covers one of the widest dynamic size ranges on the market, so 10 nanometers all the way up to 5 millimeters. And of course, this is application dependent, but the LA960 is able to achieve this measurement range because it is equipped with dual lasers. And these lasers have been known to last for a very, very long time. It also comes with a variety of different accessories that I'm not showing here, but that allows you to measure your particles for various applications, from toothpaste to chocolate to battery material to other metal oxides. Or I've also have seen people using laser diffraction to look at adjuvants or coronavirus. If you are more of a visual person, here's an overview of all the systems that we carry. As you can see, there is quite a bit of overlap, and so that kind of leads me to the next slide. When you have an overlap, how do you pick the right technique? And this is more of a loaded question, but I want to show you that whether you're using laser diffraction, the LA960, dynamic light scattering, or the simultaneous multi-laser NTA, these techniques can come to an agreement and give you a similar result if the sample is relatively uniform. And we actually encourage using an orthogonal technique to confirm your result as well. So I'm going to go ahead and pass the ball back to Young.
Great, thanks. Yeah, so move on to my examples for today. I do have quite a few of them, so hopefully I can go through them quickly. As Julie mentioned, so almost all of the example here, we did use the laser diffraction for particle size characterization. So the first is nanoemulsion application for biopharma application. So emulsion is actually widely used not just for pharmaceutical industry, but also other industries, including nutraceutical, cosmetic, and chemicals. So in case you're not familiar with what's our emulsion or nanoemulsion, it's basically formed by two or at least two immiscible liquid with one dispersed in the other. So it can be an oil in water emulsion or water in oil emulsion, depending on which is the dispersed phase or which is the continuous phase. In the case of oil in water emulsion, the oil is dispersed in the continuous phase, which is water or aqueous phase. That's I would say probably the most utilized emulsion formulation compared to other types of emulsion. And you can also form multiple emulsions, which means you can have a oil in water in oil or water in oil in water, which has multiple layers of emulsion. So regardless of what emulsion you want to produce or work with, it's important that the formulation needs to contain some sort of surfactant or emulsifier. So the function of the surfactant or emulsifier to help reduce surface tension and also to stabilize the droplet. Otherwise, although even the processing equipment is powerful enough to produce nano-sized droplets, and they will come right back together because there's nothing to separate between different droplets. I also want to mention that nanoemulsion it's different than microemulsion, although they're similar in size. Nanoemulsions are not thermodynamically stable, but they're rather kinetically stable, which means they usually undergo through, will go through some instability mechanisms that eventually can lead to stability issue. So one of the mechanism for which is important for nanoemulsion is the so-called oscillate ripening phenomenon, where if you have larger particles or droplets, that droplets will grow at the expense of the small droplets, and so it will basically absorb the small droplets and grow larger and larger. Eventually, will lead to phase separation of the emulsion. So it's important if you're creating an emulsion, you want to avoid or you want to eliminate the existence of the larger droplets. For example, anything size around a micron or larger, you do not want them to have a stable emulsion. So quickly here for why people are interested in using oil and water emulsion, because at least for pharmaceutical applications or for any other industries, is the when the active ingredients are not water soluble, which is true for biopharma application. The majority of the active pharmaceutical ingredients are actually insoluble or has poor solubility in water. So a way to help deliver them and increase their efficacy or availability is to by forming them into an oil and water emulsion. So here the oil is a carrier for the active ingredients, and by reducing the size, they can be better absorbed and achieve higher efficacy. Some of the formulation you have heard about vaccines actually can also benefit from oil and water emulsion, because certain formulation have been confirmed that has adjuvant effect. The adjuvant is a component of vaccine that helps to boost the immune response. So in the case using the oil water as vaccine adjuvant, it not only carries the antigens to the patient, but also help to boost that the patient's self immune response to the antigen, which will be the case I'm going to talk about. But before that, I just want to run a quick animation to walk you through a simple step, like how you will prepare emulsion or something similar with a microfluidizer processor. So let me go back to replay this video animation. So you will start with a coarse emulsion by mixing your oil and the aqueous phase or water phase, using some high shear mixer to make a coarse emulsion. It doesn't need to be super small in terms of size. It just needs to be stable enough before you process through the microfluidizer processor. And once you process through, you will typically get emulsion with small particle size or droplet size. So we usually call when you process everything through the system once and you collect it at the outlet, that's a one pass. So in many formulations, you may have to do one pass may not be enough. You may have to do multiple passes. Some of the parameters that can be adjusted from the processing side are process pressure, type and size of injection chamber. As I mentioned, we have different types, and the channels comes with different sizes as well. And temperature sometimes can be an important parameter for processing emulsions and other formulations. And I've mentioned number of passes. I also want to make a note here by emphasize that although the system is great and that there are some parameters you can adjust to help achieve the desired outcome, it is equally important that you have a good formulation. That's sometimes people forget about that. They rely on the processing equipment, which is true. We can help in most of the time, but you do need to have a good formulation that have the right chemistry to help achieve the processing results. So the case study here for pharmaceutical nanoemulsion is a vaccine adjuvant. This is actually a study we collaborated with our customer AAHI. So they were previously known as IDRI, which is short for Infectious Disease Research Institute. So IDRI developed a squalene-based oil and water emulsion to be used as vaccine adjuvant. So they were working on the project to get ready for a scale up. their process to be prepared for in case of a pandemic response. And so the goal for that scale up is to be able to manufacture enough dosage, which in this case 50 million total dosage of vaccine or adjuvant within 3 months of time period. So they did their calculation that to be able to meet that, on average, they need to produce 1 million doses every day. And obviously, the requirement for accepting emulsion or some of them are listed here. So the particle size needs to be the average particle size needs to be around or less than 100 nanometers. There's some other characterizations about the OU and excipient concentrations, homogeneous visual appearance, and etc. And since this is a vaccine, it needs to be injected into patient or people. As I mentioned before, the final product needs to be able to pass through a filter for sterilization. That's why they're looking for that particle size, and they need a tight distribution. And lastly, this is a vaccine product, so it needs to be manufactured at a GMP facility. So when it started, they initially developed the process and the formulation with a relatively lower oil concentration, around 4%. And the process needs to run the material or the product 10 times through the system to reach the target size. And so before we do any scale up test, we worked on actually optimizing the formulation and the process to help reduce the total process time, because otherwise to meet the target for the scale up in response to a pandemic attack, they may need like multiple machines and running that for a longer period of time. So after optimization process, we were able to optimize both formulation and process. You can see here we're able to process a more concentrated emulsion with 30% of oil, and only require 2 passes to achieve the target particle size. And if you do the math, and this overall optimized process can reduce the amount of the time from the initial process by 97%. So the final optimized, it only needs 3% of the time to process that same or to produce the equal amount of dosage. And here's the the particle size after optimization. You can see the results from one of our pilot system, which is the M110EH Microfluidizer processor. You can see the particle size has been reduced greatly after the first pass, and after the second pass, it made requirement. If you do need a little smaller particle size, you can run another pass, but which is not important in this case. And when we take this process to scale it up from our pilot scale to the production scale, which in this case we use our M7250-30 in fully enhanced machine. It's a 50 horsepower machine, can deliver a flow up close to 5 liters per minute at the maximum pressure of 30,000 psi. You can see at the results shows here, the production scale results matches the pilot scale results very well. So the first pass, we actually produced a slightly better particle size or smaller particle size. But the second and the third, the size are almost identical. You can see the curves are overlapping. So this demonstrates excellent scalability of the Microfluidizer technology. And back to their process, with the final optimized process and improved in other areas, they were able to achieve efficiency and increase the production capacity to 5 million doses per day. So that's 5 times of what they initially anticipated or calculated daily dosage requirement. And all of this 5 million doses can be processed with just a 200-liter batch size, and the total processing time, including pre-mixing and processing through our system and post-process filtration step, can be done within an 8-hour time frame, which is a single shift. Yeah, now I'll switch to my other applications. So first, I will like to talk about applications for cosmetic products. So in cosmetic products, popular formulations are including emulsions, which is widely used in many of the products, including perfumes, shampoos, lotions, and creams. So there are different formulations, different textures, but they basically are all formed with emulsions. There are other formulations, include liposomal formulations, which is also very common. Liposomes have a benefit of delivering both or encapsulating both hydrophilic and hydrophobic actives. So it's very flexible. Listed a few different active ingredients can be encapsulated here. And there are also a few other dispersion-based formulations are used in cosmetic products. And an interesting topic here, I will give you an example, is the self-disruption can be utilized in cosmetic formulations or products as well. But first, let's look at an example for emulsion. So here, it's an emulsion for alcohol-free perfume. I think we all know that most perfumes available right now are alcohol-based because alcohol is often the main ingredient in the composition as a solvent. Since the aromatic compounds to make the perfume usually are not water soluble, so they need to be dissolved. The alcohol is commonly used, and there's some drawbacks when you use a large amount of alcohol in the product, especially as for cosmetic products. In the case of perfume, that alcohol can cause skin dehydration and skin barrier damage, and this is especially important for sensitive skin. So there's a need to drive or to ask for alcohol-free formulation. There are some trends here, such as the clean beauty movement. They'll try to push us to remove solvent from the product. With that being said, so the perfume companies are now looking at alternative ways to make perfumes, hopefully to use water-based alcohol-free formulations. That being said, I've mentioned before, using the emulsions is probably the best approach because now you have the aromatic compounds that are not water soluble. If you don't want to use solvent, you can try to use an oil as a carrier and then formulate that into a nanoemulsion. By doing that, it also helps to achieve similar appearance, especially the translucency appearance of the product, because most perfume products are like clear or translucent. So customer getting used to that. So it's good to sort of keep that as the alternative product, and if that's important. And obviously, reducing the droplet size will help achieve that goal as well, because we know that when the droplet size getting smaller and smaller, it starts to allow light to pass or get around of the particles and help to improve that appearance to translucent or transparent. So a quick example showing here is a perfume formulation or emulsion formulation processed using our system. The goal here is to create a nanoemulsion encapsulating fragrance and achieve a mean particle size around 100 nanometers. So you can see we can quickly reduce, as both shown in the table with the particle size and the distribution curve here. And our processes are efficient, quickly reduce the particle size from a large and processed material, which has an average size a little less than 10 microns to a couple hundred nanometers just after a single pass. An additional pass keep reducing the particle size. After 2 passes, we're a little over 100 nanometers, about 130, and after 5, we reached our target with the particle size, average size slightly over 100 nanometers. So you can see here the laser diffraction, we're able to capture both the larger unprocessed particle size and the smaller processed nanoemulsions. So the next example for emulsion is actually a shampoo emulsion formulation. So in this case, it's a bond treatment shampoo. Hair bonds are responsible for the shape, strength, elasticity, and shine of the hair. So if you have broken bonds, that can lead to brittle, damaged, and eventually broken hair strength. So here, the goal is to process a special shampoo that's formulated to help regenerate bonds and to repair damaged hair. And the goal is to achieve produce a uniform and stable emulsion with D50 particle size less than 200 nanometers and D90 particle size less than 300 nanometers. In case you're not familiar with the terminology here, I forgot to explain earlier on. So the D10, D50, D90 represents the percentage of the particles that are less than that size. For example, D50 represents the 50% of the particles are less than this size. Depending on the method of analysis, it can be either volume, for example, that's for a laser diffraction based method, or intensity with the DLS measurement. So again, you can see from the particle size data here, it shows the very quick reduction of the particle size. In this case, it took only one pass to reduce the particles from about 5 microns with D50 to 130 nanometers, and even the D90 particle size is less than 200 nanometers. So we were able to achieve the target here with just one pass, which in this case is excellent results, because the lesser the pass, the more product can be processed through a processor, which is ideal in this case. So the next formulation is actually a liposomal formulation. So in this case is the botulinum toxin. I think it's better known as Botox. So it is a highly potent neurotoxic protein. So in cosmetic applications, it is used for reducing facial wrinkles and by relaxing the facial muscles underneath after injection. So the goal here is to formulate this into a liposomal formulation, because for transdermal delivery, the target particle size is less than 200 nanometers. Again, you can see the before and after from a big size unprocessed product material to a processed with a D90 less than 150 nanometers, so well below the target size. And the size also are confirmed by microscope images showed to the right here. You can see visible large particles before processing and non-visibles after processing due to the limitation of the microscope used here. We're not able to see nano-sized particles, so that at least tells you there's no any particles in the micro range. Now, switching gear here, I mentioned about cell disruptions. So I want to tell you a bit more here. Cell ruptures are actually widely used in pharma and biotechnology industries, but they're also used in many other industries because cells are very popular factories. Actually, can be used for producing engineered or recombinant products such as proteins, enzymes, and other products. They have a long history of over 50 years that people have discovered and utilized cells for making the products. Cell rupture is not always required. So it is only required when the products of interest are not expressed extracellularly or in other ways, it's not secreted by cells. You need to rupture the cell membrane to harvest the intracellular product, or if the product in the cell membrane, you have to harvest the membrane fragments and do additional purification steps. But in any case, in cell rupture will be required to help extract the product. There are many different types of cells can be used. I would say most commonly used are either bacterial cells. E. coli is probably one of the most popular one, and also some of the yeast cells, which are good for synthesize some of the recombinant proteins and enzymes. A variety of cells are also used in cell-based cultured food or nutraceutical products. With the emergence of the next generation therapy, such as gene therapy, there are applications for using mammalian cells to grow and harvest viruses which are helping deliver the genes to the patient. So here in this case, commonly used virus is adeno-associated virus. So the virus will be engineered to carry a gene that can either correct or edit or insert or knock off a faulty gene or missing gene in the patient. So once the virus is engineered, it will grow in mammalian cells. So most commonly used cells are either HEK cells or CHO cells. So once they grow into a certain number, they won't come out of the cell. So we need to rupture the cell and to release the viruses. So that's the application for using mammalian cells for virus replication. And there are also many different types of algae cells. Initially, people are looking at them to generate biofuels, but I believe now algae cells are also being studied and researched for producing many other different type of products. But regardless of what cells you're working with or you want to work with, and the choice of, you need to look at how to efficiently rupture them, because each cell types will require different forces to rupture the cell membrane. As shown here, a mammalian cell is on the lower end. They only need very gentle conditions and low shear to rupture them. Bacteria is in the middle, and yeast cells are among some of the tougher cells, which will usually require the highest level of shear to rupture them. And algae, depending on the species, they can either use medium high or high shear rates. So the point here is you just want to apply, because a lot of the products you want to harvest from the cells are sensitive, so you want to just use enough force to open them and not to damage the product. So which comes to the benefit with using Microfluidizer technology. We can pinpoint by adjusting the pressure and selecting of the chamber to pinpoint the shear forces, then our uniform processing can be applied to just the rupture enough the cells and not damage any of the product. So I know I'm running out of the time, so I'm trying to speed up here. So here, this is an example for rupturing bacterial cells, and the cell of interest here is a bacillus cells, and they are a gram-positive rod-shaped cell, and you can see from the image here. And they also form spores. So I'm not sure all of the spores are exhibiting this character, but some of the spores can resist to very harsh environmental stresses, including UV light exposure, which made them a potential candidate for formulating into cosmetic applications such as sunscreens. So obviously here you need to rupture the cell to release the spores. And you can see from the results here, the particle size analysis clearly showed the size difference between the unprocessed and the processed. Although in this case, the cells before rupture, the intact cells are rod-shaped like, they will not be measured accurately by typical particle size analysis method, because most of the method assume the particles are spherical. This is the rod-shaped, but they can still give you a directional trend that indicating at a very quick check for the outcome of the product or the process. So you can see the particle size decrease a lot, which means the most of the cells are ruptured into smaller fragments. And you can also see from the microscope images here, before and after, that most cells were ruptured. Okay, so move on to energy performance storage application. So the demand for energy storage devices are increasing because of the increasing interest or increasing generation of alternative, especially clean and renewable energy. So some of the common devices are batteries, supercapacitors, and fuel cells. Carbon-based materials have gained extensive interest with their unique properties in terms of electrical, thermal, and mechanical properties. Some of the commonly used carbon-based materials are activated carbon, carbon nanotubes, graphite or graphenes, or quantum dots, sometimes also called carbon dots. So the Microfluidizer system actually works for all these different carbon-based materials, but my focus today will be activated carbon and carbon nanotubes. So for activated carbon, it's used in fuel cells. So fuel cells are devices to convert chemical energy to electricity. Most used fuels are hydrogen. It can be used in a wide range of different applications across multiple sectors, including transportation, buildings, and long-term energy storage. It compared to conventional combustion-based technology, it offers much higher efficiency with zero or low emissions. So the PEM, which is the proton exchange membrane, or also known as polymer electrolyte membrane fuel cells, are one of the leading technologies that people are interested. I'm not going to explain how fuel cell works, but I want to point out, look at the structure here. The fuel cell employ an anode and a cathode, and there are catalyst layers on each side using the same catalyst. So the catalyst is responsible for chemical reactions. On the anode side, it splits the hydrogen molecule, and on the other side, it forms water through the oxygen reduction reaction. So the catalysts used are usually precious metals like platinum or some other precious metals, and they are supported by activated carbon. So the idea here is you want to increase the efficiency of the chemical reaction. You need to increase the exposure of the catalyst to the fuel or to oxygen on the other side. So activated carbon has large surface area, even though you have to still disperse that very well to help increase that surface area and to have the catalyst exposed to available for reaction. And on top of that, as I mentioned, they're precious metals, so they're very expensive. So by achieving a good dispersion, you can also use less ink for the catalyst layer and save costs on that side too. And so here's an example of processing that ink dispersion. So the goal here is to achieve a homogeneous dispersion with particle size below 1 micron. And you can see from the image here and the particle size that we were able to efficiently reduce that size to achieve a particle size far below the target particle size. On the other side of the other energy storage devices, we're talking about lithium-ion batteries or supercapacitors. So they both use electrodes, and the electrodes are the costly part, at least for the batteries. And there are some challenges for materials that are currently used to make the electrodes. And so adding carbon nanotubes is a promising idea to help improve the power energy storage and life cycle of the electrode design. So you can see that the dispersed nanotubes will help that. On the supercapacitor side, it's a similar storage to help improve the efficiency. The high specific surface area are important, which all requires that very well dispersed or deaggregated carbon-based formulation. A quick example showing here with dispersing carbon nanotubes. So the goal here is to reduce particle size to below 3 microns with a relatively low fluid viscosity. Again, so carbon nanotubes has a long aspect ratio, so they won't be measured accurately, but at least as I mentioned, the particle size analysis is a very quick check, and to at least qualitatively show the processing results. You can see the particle size reduced beautifully from the end process after 1 pass, and after 5 passes, the mean particle size was less than 3 microns. But if you need further reduce the particle size, additional passes will help. After 10, most of the particles are less than 2 microns. Okay, I have 2 other final examples I want to walk through quickly. So the first is the flavor emulsion. So flavor emulsions are water-based and usually highly concentrated flavors. So it's different than a flavor extract, which usually contain alcohol to help dissolve the flavor ingredients. And To make a flavor emulsion, it's very flexible. You can make with many different flavors and colors and styles, and it can be used in both beverage or a variety of edible products. So again, in this case, you want to achieve a small and uniform particle size to ensure the stability of the product as well as achieve desired appearance. So example here is to produce a stable flavor emulsion. You can see the process outcome from just one pass through a microfluidizer processor from a very large unprocessed emulsion of size close to 70 microns to beautiful, very sharp peak here representing a very narrow distribution, uniform distribution around 300 nanometers was successfully achieved only after one pass through the system. So my final example here is another nutraceutical emulsion, which is a emulsion developed for CoQ10. So I will not read everything here. So CoQ10 is not an ingredient approved by FDA to treat any medical condition, but it is a powerful antioxidant, which is widely used as dietary supplement in some cases in cosmetic products. So it is not water soluble, it's instead of fat soluble. So formulating into oil water nanoemulsion helps to increase the bioavailability of CoQ10. Again, you want to achieve a good size and with narrow distribution. And as I've shown here, again, the Microfluidizer technology is super efficient to reduce the particle size from the large unprocessed emulsion to smaller size. And even one pass, we were down to 150 nanometers, and 2 or 3 passes continue decrease the particle size to further. So, and my final slides here, just to quickly show you a selection of our product. We have a range of lab scale machine, then we have a small pallet, a larger pallet, all the way to production scale machine. For this one lab unit model and all the larger scale machines, we also have GMP capable models. And apologize for running over the time. And with that, I'd like to thank everyone for your attention.
Thank you so much, Yang. So I know we have quite a few questions that came in, but because we want to honor everyone's time and also Yang's time, you can follow up any of your questions by sending an email to labinfo@horiba.com. So I think we have time for maybe one question. Are you ready?
Yep.
Okay. So does the microfluidizer have temperature control?
The quick answer is yes. So I showed there's usually a moderate temperature increase inside the induction chamber because you're converting pressure energy. It will dissipate to heat. That's why we have temperature control device right after the induction chamber. On the smaller lab scale machine, we have cooling coils. On the larger system, we have heat exchanger. And with the larger system, we also have capability of design a closed loop temperature control capability for our customers.
Thank you. And if this is really quick, there's one more question thank you so much for that useful information. Can you please talk a little bit about how cell disruption maps into the particle size distribution space?
Yeah, like I said, I don't think, measuring particle size will be super accurate in the case of cell disruption. But you can get an idea, like on the intact cells, they're usually large in the micron range. And once we break the cells, actually we tear the cell membranes into many smaller pieces. And so you can certainly monitor that particle size. And it can be actually used as a trend or indicator. I guess depending on your product, you can sort of develop as a quick quality check. But obviously, it's most important to analyze the product you actually harvest from the cell by doing cell rupture.
All right. Thank you so much, Jan, for the excellent talk. As you can see, there's so much to unpack using the microfluidizing technology, as well as using the HORIBA laser diffraction unit to cross-check or to provide feedback loop. And so we can dive straight into your specific application if you just contact us. For now, I want to thank everyone for your time and for your attention. Make sure you join our newsletter or connect with us on LinkedIn. Is there anything else you want to add before we wrap?
No, yeah, thanks for organizing the webinar, and thanks everyone again for attending. Apologize for running over the time again. We have our contact information listed here on the slide, so feel free to reach out to either Julie and I or both with any further questions.
Absolutely. Thank you so much, and bye for now.